regarding a very interesting topic, which is CRISPR technology, its application in agriculture and human health. So, before going into the topic and discussion from the side of Dr. Sanjay Ghosh, it is my immense pleasure to introduce you Dr. Sanjay Ghosh, who is an eminent scientist, a great researcher in this field itself. Um, if any one of you have previously attended the talks given by Dr. Sanjay Ghosh, you might have really enjoyed the talk, the way he present his topic, the way he communicate with the students and the participants of these online talks. It is really commendable. And sir, I am a big fan of you regarding this topic. So it is again, as I said, my pleasure to introduce you on this topic of the CRISPR technology. And for the participants who have joined us, let me introduce you, Dr. Sanjay Ghosh. Sanjay Ghosh, sir, he is a alumnus from the BHU. He has carried out his MSc in zoology from BHU, and then he moved to CSIR IICB Kolkata for his PhD. After that, he has obtained a lot of experience, research experience from various institutes, which are actually the dreams of the many researchers, like EMBL, which is in Germany. Then he has also did his postdoc from the McGill University, University of Cambridge, and also from the Oxford University. So all these names, these are very pioneers in the field of biotechnology, very pioneers in the field of life. I hope everyone is aware of all these institute, all these universities. So sir is having immense experience in this field of CRISPR. Currently, he is associated with IBAB, which is an institute of bioinformatics and applied biotechnology, which is again a premier institute, research institute situated at Bangalore. So, sir, now I would hand over all participants to you so that we may start the talk and they may get benefited from your experience, from your the simplest way of explaining the complex thing. That is what I can describe you. So, sir, now everything is up to you. Sir, you are on mute.
Sorry.
Yeah.
So thanks a lot, Poonam madam, for a very kind introduction. So I'm sure that you all will have the opportunity to go where I have been. So this is a very good platform to discuss many frontier areas of biology and biotechnology, and we all here are to learn from each other and also discuss the latest topics, right? So I hope I can interest you in this topic. My lab does work on CRISPR and various types of CRISPR that we do, but today I will be talking about the applications of CRISPR.
Sir, just a minute to interrupt you. Gopalji sir, he has also joined this meeting. So
Okay.
actually, this concept of science dialogue series, it is actually the brainchild of Dr. Gopalji sir. So just a kind of acknowledgement to his effort. I am here to conduct or coordinate this particular event. Otherwise, this complete uh, talk series is a brainchild of Gopalji sir. He is always there to help the students and communicate with the students in different fields of science. So, Gopalji sir, this is a welcome for you also. You are in this meet. And now, sir, I just request you to continue and start the talk.
Okay. So thanks again. So I will just share my slide and I may switch off my camera just for bandwidth issues. So please bear with me. Uh, just a second. Ah, okay. So I hope you all can see the slide.
Yes, sir. It is visible to us.
Okay, fantastic. So now it is full screen, right?
Yes, sir. Yes, sir. Sure.
Okay, so welcome everyone. And as Poonam madam was saying, that this is a very nice opportunity that Gopalji sir has created, and we all should make the best use of it. So without further ado, I will start with the discussion or the topic that I have chosen today. And it is to do with the CRISPR technology, how it is applied to solve problems or to make better agricultural as well as human health products. So before going into the application per se, I mean, of course, all of you have heard about CRISPR. I just wanted to take a few minutes to first introduce that how CRISPR-Cas9 system was identified in the first place as a simple biological problem, and then later on it was used as a genetic tool to modify, to edit, to delete, and so on of different DNA and RNA sequences as we know today. So CRISPR-Cas9 system, as you know, is basically an adaptive immune system of the bacteria. So what happens when the bacteria gets attacked by a bacteriophage? So the bacteriophage injects its DNA and then it multiplies and it lyses, it comes out. That's all we know, and that is the standard textbook material. But what some bacteria have evolved is that they take, I'll take the laser pointer maybe, and then that will be good. So what some bacteria have evolved is something called a CRISPR array. That means there are very conserved sequences which are flanked by non-conserved sequences. These different color codes means these are different sequences. And they are numbered from 1 to 10 here, but it can be longer also. Now, what are those sequences which are colored here? They are nothing but pieces of DNA around 20 to 22 nucleotide long or base pair long, which are taken from these phages which attack the bacteria. Now, why they should do that? Any foreign DNA that enters a cell is actually a very dangerous sign for a cell, because every cell will like to protect its genome, and bacteriophage or even plasmid DNA that we use to transform, they are foreign. So what bacteria has evolved a mechanism to cut out a piece of that DNA and integrate within its genome, and that forms a memory of that bacteriophage infection or even a foreign plasmid inside the DNA, right? And this memory is close together with another protein coding locus called CAS. So we have to remember there is a CRISPR array containing some conserved sequences and interrupted by this so-called this memory of the foreign DNA attacks. So that is called acquisition, the first step that happens within a normal bacteria. And when this and then this acquisition is followed by a step called expression, where this array is expressed by RNA polymerase. You get a long concatemer of an RNA which has these different colored regions in between, but this constant region forms a very conserved stem loop structure, and this is called a pre-CRISPR RNA. At the same time, from this Cas locus, this protein coding gene. So you have to remember that this CRISPR array is non-coding. There is no protein coding gene. However, very close to it, there are genes which are protein coding, and they are the Cas proteins. So these Cas proteins, they are independently expressed, and along with the CRISPR array, this RNA and this protein forms a complex. There are some proteins within the Cas locus, which is RNA's enzymes, which will cleave this into separate pieces, and then they will form a complex, and they will keep it inside the cell for surveillance. Now, whenever the same bacteriophage attacks again and injects its DNA, this Cas guide RNA, so-called this CRISPR RNA complex, will identify that foreign DNA and simply cleave, will introduce a double-stranded break. And this can not only happen because of an infection by the same bacteriophage, but also it happens if you have a plasmid DNA. So whatever memory it is kept, that memory is actually transcribed, and together with a protein forms a complex. That complex is floating around in the cell, and whenever there is a same DNA comes in, it forms a complementary base pair and goes and chews it up. And we know that cleaved DNA cannot survive within a cell. There are exonucleases which will destroy it. So this is the background of the CRISPR, which naturally occurs in prokaryotes and archaea, which is lower eukaryotes. And now recently has been found something similar in eukaryotes, which I will not talk about today. So this CRISPR it stands for, as you know, clustered regularly interspace short palindromic repeats. So these are clustered, this array, and they are having fixed length in between them. They are very short sequences and they are also palindromic so that they can form a stem loop structure, right? And this is the full form of CRISPR array. Now, these are the proteins which are right next to it, which are essential for this degradation pathway. These proteins always complex with this CRISPR RNA, and that's why they are called CRISPR associated. In short, CAS. So that means this CAS and the CRISPR, they are coming from the same adaptive immune system. So, to make things very simple, what scientists realize that if it cleaves a DNA, then we can use it to edit genomes. So what they did is that they cloned this Cas protein, they cloned this RNA part from the bacteria, and then they developed something we called as an RNA guided DNA endonuclease. Because now we need to express a protein which is the Cas9 nuclease, which was first discovered. We need to express a guide RNA having a unique 20 nucleotide targeting sequence which will target any DNA sequence that you want, except that that sequence should be having a PAM, which is called a proto spacer adjacent motif, right? So a PAM sequence is needed. If it satisfies this condition, you can literally cleave any DNA at any place based on what you engineer here. This 20 nucleotide is unique, right? So it is a very simple system, two component system, which depends on the guide RNA, a PAM sequence, and a nuclease. That's all is needed. And this is the structure of this so-called guide RNA, which is conserved. And this is the spacer, which I showed you in different colors in the earlier slide. And this is what is the targeting sequence. And this has to be 20 nucleotides for Cas9. So 20 plus roughly 90 here gives the entire guide RNA structure, and this stem loop structure is very, very conserved. Now, what does this CRISPR-Cas9 do? It's very simple. There is nothing big deal about it. CRISPR-Cas9 is used for engineering DNA just by creating a double strand break. So all it does, it will go and bind here, and then there are two nuclease domains which will cleave the phosphodiester bond on each strand of the double strand DNA. Now, if it cleaves the DNA in like single cleavage on each strand, you end up generating a double strand break. Now, double strand break in a cell is a bad news. So what will happen is that the entire job of CRISPR is just to create a double strand break in a sequence that you program. Once you have a double strand break, the job of CRISPR is over. The cellular DNA repair machinery will kick in. And what it does is that there are two different ways to repair this DNA, which is a double strand break now. So one is called a non-homologous end joining, which is very imprecise. So it randomly puts bases here, or there is a precise way of joining it, provided there is a donor template, and that is called homology repair or homology directed repair. So this property of DNA repair pathway, which is triggered by the Cas proteins, is used for editing genome. So that is all one has to understand, that if we cut the DNA and if it goes through the NHEJ pathway, there will be random bases inserted or deleted. And that's what they are called indels, right? The in coming from here and del coming from deletion. And if you do it in the coding region of a gene, then, and it is a random process, you may delete or insert nucleotides which are not a multiple of 3. And 3, why? Because codon, right? One codon is 3 nucleotides. So if you have now deleted 2, you basically have changed the reading frame, and thus there will be a stop codon somewhere. And that is how you create knockout of the genes. Now, if you want to do a precise editing, because this is quite imprecise, but it ends up killing the protein function. But if one wants to do a precise way of editing genes, then you have to provide a donor DNA. And this donor DNA should have the flanking regions cloned, and in between one can put whatever sequence they want. And this is what has been used for gene modifications, that means precisely. And using this feature, you can correct diseases, and we'll see some examples of that. in not only you can actually correct a given amino acid codon with a correct amino acid this codon, but you can actually put large chunks of DNA also inside once a double strand DNA break has happened. You can put a new gene, you can put any sequence that you want, thereby tagging a gene with fluorescent proteins or even fusing genes or making a in vivo fusion, right? So this is the property that is used. So you have to remember that one thing that stands out, that CRISPR involves editing of endogenous DNA. There is no foreign DNA that is expressed. There is no foreign DNA you are editing. You are editing the DNA present in the nucleus of that cell, right? There is nothing foreign coming. So to summarize, basically that means CRISPR Cas9 can be like a Swiss Army knife. Like it has so many blades in a normal Swiss Army knife. So you can do integration, you can do point mutation, you can do deletion, and you can change bases literally at will, as long as you have the PAM sequence and you have a targeting 20 nucleotide sequence, right? So now, if you look at the application, so there are now currently a lot of FDA-approved CRISPR therapies are in clinical trials, and they kind of target different parts of the body of different diseases. For example, in soon when the SARS-CoV-2 this pandemic happened in May 2020, a year later, there was a CRISPR-based therapy to basically stop the viral infection from infecting the lung tissue, because it is lung specific because of ACE2 receptor. So there are a bunch of other things that is going on from 2017 till even recently, till last year November, for the SCID and for other metabolic disorders. FDA has approved that one can do so-called gene therapy, that means correct the gene or edit the gene based on CRISPR technology. So now how it is done or how it can be achieved. So there are two broad ways of doing it for humans. So one is called an ex vivo approach and one is an in vivo approach. That means I can take the guide RNA and the Cas9 protein, I can make a complex in a test tube, and then I will load it into a nanoparticle or some kind of delivery vehicle, and I'll inject in the blood. Now this thing will go throughout the blood. It will finally end up in, if it is targeted to liver, then it will give to a specific organ, and then those corrected genes will now be repaired in that particular organ, thus maybe removing the disease phenotype. The other way of doing it is an ex vivo. That means we can take out the cells from the patient, we can edit those genes outside in a petri dish, and then after editing, we can select the healthy cells and we can push it back to the individual. So in one case, we are injecting the DNA directly in the blood, and in the other case, we are manipulating the cells outside of the body and then we are putting it back. So ex vivo, in vivo. And this is what you have to just remember.
Sir,
Yes.
sir, just question, sir, just a one question. Sir, if we are injecting such kind of nanoparticles, aren't they considered as non-self and non-attacked by the immune system of the person?
Yeah, so one has to choose the nanoparticle appropriately that will not be seen as foreign, right? So these are usually inert materials which are coated with certain chemicals where it doesn't usually, invoke an immune response. So
Do
that
we
has to be
do
mature.
we also place some kind of cell markers on these?
Take it to the liver and will attach there and will be, kind of targeting there. If I want to target to brain, then something specific to brain should be in that nanoparticle so as to target and be more specific therapy.
Are these particles are capable to cross the brain-blood barrier?
No, most of them don't. Blood brain barrier is a very tricky thing. That usually they won't. But there are now some reports where people use exosomes and other kind of vehicles where
Okay.
they can target the brain cells, and it has been reported to cross the blood brain barrier.
Okay, sir.
Yes. So, so this is ex vivo and in vivo approach, right? So one of the examples that I want to give is something called CAR T cell therapy. You have heard about it, maybe. There is recently Nobel Prize given for immunotherapy. So one of the things of the immunotherapy is so-called the T cell therapy, and they are called CARD because it is chimeric antigen receptor, right? So that is a synthetic receptor. The cell doesn't have it; you engineer it. So how do you do it? currently the pipeline is that you draw the blood from the patient, you isolate the T cells, and then you edit those T cells using CRISPR technology. And you can insert this chimeric antigen receptor using CRISPR, integrate within the T cell genome because in the DNA, and you can also delete some other genes, and I will show you in after a few minutes which genes are deleted and why. So you can delete some genes. Basically you modified and engineered this cell outside in a petri dish, and then you take this edited cell, you selected the cells which have been edited from the ones which have not been edited, and then you put it in this like a bag, and you infuse it within the patient. So now these T cells will go and will express a certain antigen, a receptor, which is actually selected to bind to a tumor antigen. And that is one of the therapies that people are trying to do for cancer therapy, right? For solid tumors. Although CAR T cell therapy also has been used for the blood cancers, but there is a challenge currently to do it for the solid tumors. So the idea is that these T cells will go now, they will have a extra receptor that we have engineered, and now it will go and kill the cancer cells. So this is one of the ways of targeting the cancer by manipulating the T cell of the patient itself. So there is no immune response or unfavorable immune response there. So that is one of the advantages of that. Now, what are the other examples where the CRISPR has been used to modify the human genome, right? So this was a paper which came out in 2015 and caused a lot of controversy, because in China, a group of Chinese scientists, they use CRISPR Cas9 in the pre-implantation embryos. Of course, these embryos would have never, gone beyond 16 cell stage. They use this zygote to correct this beta hemoglobin gene, and the idea was that can we edit the gene of this beta globin in humans so that they can be cured of thalassemia or even sickle cell anemia later on, as we'll see. So they did that, and they showed that a fertilized human embryo, one can throw in DNA like CRISPR Cas, as well as the guide RNA, which targets the beta globin gene, and they could edit. So that was first proof of concept. But since this is done in a germline, which is banned and forbidden, this work was not followed up later on. But now, recently, you must have heard, right, in November 2023, just a few months back, UK was the first nation to approve CRISPR-based therapy. And this was to target sickle cell and thalassemia. And this has been approved for use in humans. Soon afterwards, in December, FDA also approved the same gene therapy based on CRISPR for sickle cell disease. So all of you know that the sickle cell is caused due to a single amino acid mutation. Thalassemia is the same, that the hemoglobin chains are not formed. So these patients, they suffer for because of the sickle cell, the blood cells, they get clogged in the capillaries. It's painful, as well as the oxygen carrying capacity is less, right? So these patients need, usually the thalassemia patients, depending on the condition, blood transfusions to lead a normal life. So what is this CRISPR based therapy that they developed? So just to tell you that we had, during the development in the fetus, we have a form of hemoglobin that is expressed in our blood, and that is called hemoglobin F. And this F has two alpha chains and two gamma chains. But when like we are born, this gamma chain is replaced by a beta chain. So now the adult hemoglobin, which is called HbA, A for adult, consists of two alpha chain and two beta chains. So that means that there is a switch from gamma to beta. But this is important. Nature has evolved this. Why? Because this gamma hemoglobin, containing hemoglobin, can carry more oxygen. And when it is in, like a fetus is within the mother, then it needs to capture as much oxygen from the placenta. But when we are outside in the environment, we don't need to carry so much of this oxygen normally. So this beta chain has a lower oxygen carrying capacity, right? So this is how the switch happens. But what is happening is that when the beta chain is to be expressed, this gamma should not be expressed. And for that, there is a gene called BCL11A. This BCL11A is a repressor. It represses the expression of the gamma, and so that the beta is only present in adults. So now people who have this sickle cell anemia, they have anyway lower oxygen carrying capacity because of this beta hemoglobin. What this treatment involves is just using CRISPR to delete this gene. So if you remove the repressor, then gamma will be made in these adult people. And so therefore, and since gamma has a higher oxygen carrying capacity, this lower oxygen capacity phenotype of the sickle cell patients will be much cured. I mean, it will not be normal, but it will be much better than what it was. So this is all simple one gene knockout using CRISPR that has currently been approved for therapy in humans in UK and US. So this is the simple way of using CRISPR to knock out genes. Now, what is also work is ongoing is that can we also cure or treat HIV, right? So HIV we know is a retrovirus. The genome goes and integrates within the genome of the humans, and it lays dormant, and then it gets expressed and it causes diseases at times, right? So latency is very dangerous to treat, or very tricky to treat. So what they have shown in the mouse that by using a CRISPR, you can design to guide RNAs which will cleave this retrovirus, this HIV genome, which is present in the human genome itself. So once you cleave, then this viral genome is non-functional. You can actually remove this entire genome by designing guide RNAs which flank this entire cassette, or you can cleave these coding regions and different parts of the genome so that the virus, if when it wakes up, it is non-functional. It cannot cause the disease, right? So this is a simple approach. It has been tested in mice, but in humans there are tests ongoing. Now, one of the ways of using CRISPR is also to do so-called high-throughput CRISPR screens, and this is currently a kind of a hot topic. So, what to do? I mean, it's a very simple thing to understand. So, we should have a cell line. So, this is done in petri dish, nothing in the in vivo. So, we have a cell line which will express Cas9, and these can be any kind of cell lines, primary cells, cancer cells, organoids, etc. What we have to do is to make a library of guide RNAs which will target every coding region in the human genome, let us say, right? So there may be 80,000, 90,000 guide RNAs which target every gene in the genome. So if we are looking for knocking down the entire every genome in a petri dish, we should have more than 80,000 cells, and it is designed in such a way that each cell it will be knocked out for one gene. So when we knock out these genes, of course, the cells which have knocked out the essential genes will die. Only the non-essential genes, if they're knocked down, they'll survive. And then we can ask the question, okay, which of these genes are needed, knocked out genes, will prevent virus multiplication? For example, you can ask any question, right? Or drug sensitivity. So if you challenge with a virus and if that gene product was a receptor, then this cell will not get infected with the virus anymore, and you can identify which genes was knocked out. So it is done in a genome-wide scale. So this can lead to discovery of host factors involved in a variety of pathological conditions, development of drugs, understanding how pathogens interact with the host proteins, and so on, right? So this is once you have identified those cells, you can actually do a next gen sequencing and find out which guide RNA is residing in that cell which was now refractory to virus multiplication. So that's a very straightforward approach, although it needs some technological things. But here you are doing high throughput, unbiased manner of discovering genes or the genetic basis of drug development or even pathogen introduction. So
Sir,
how it
sorry to interrupt again. Sir,
Sure.
when we are saying about these Cas9 expressing cells in bulk, this the this protein Cas9, it is expressed at the gene level, or it is some kind of plasmid or some kind of vector in which this is being expressed?
Yeah, so that's a good question. So actually, it is put in a transfection vector where it under a mammalian promoter, and it is integrated within the genome so that it is continuously expressing Cas9.
Okay.
And then you can throw in any guide RNA, it will form a complex and it will go and knock out the gene based on the targeting sequence of that guide RNA. Right? And this is happening parallelly on a petri dish in millions of cells. So that is the beauty of this. So we don't knock out one gene at a time. In
Mhm.
this case, we are knocking out thousands of genes, maybe 20,000 genes at the same time in different cells. And then we are selecting it and finding out which guide RNA knocked down which gene. So that way we can mass, identify host factors.
So these vectors are commercially available, or one has to generally do the cloning by
They
themselves.
are available from AdGene.
Okay.
Even the guide RNA library one can purchase from the AdGene. So there is a guide RNA library for human, mouse, yeast, all model organisms. Now there are guide RNAs. Like
Sir, one query was earlier also, but I was a bit hesitant to ask about it. You just took the name of East.
right,
How it is more efficient than the conventional site-specific gene tagging that the earlier slide that you have shown. Actually, in our work, we have tagged, we have deleted the gene by using those 100 base pair sequences of the site-specific location,
correct,
like homology based basically. So
correct, correct.
why we should be doing this CRISPR thing in each cell if we have a efficient system already? Just you have to synthesize the primers, then carry out a single PCR, single step PCR you have to carry out, and then you are getting the mutants which are having these kind of deletions and these kinds of tagging point. So how is it beneficial to carry out in each system?
Yeah, so in the East, as you correctly said, that the natural recombination rate is very high. But what has been seen, and we also see that usually the donut template that you throw in has to be linear. Uh, but
Yes.
but CRISPR-Cas9 allows even plasmids to go in at a very high efficiency. So the rate of recombination or the integration in the yeast genome is close to 100%. Like almost all yeast cell will have the gene. Number two, when if you are not tagging the yeast genome, you have to use a selection marker, a oxotropy marker, right?
Yes, yes, yes.
So here it is markerless. You don't have to, because the efficiency is so high that you will get your desired gene without using a selection marker. So that reduces the cargo size quite substantially.
That
So
is true for the deletion also. Or
yes, yes. So
okay.
you can delete.
But how to identify them? I
Identify.
mean to say ki how the selection part would be done if we don't have any kind of marker there inserted. Like we
So
generally used to use that kanamycin resistance G418
correct,
for
correct,
E
correct.
cells.
Correct. So here you can do genomic PCR. You have to design
Okay.
primers where the guide RNA is targeting, and you have flanking that, and you can do a PCR to find out if the modification has happened, and you can sequence it also to know that it is in frame and so on. That
Okay.
you can be done very easily.
Okay, sir. Basically, efficiency is quite high in case
Yes.
of this CRISPR.
Yes. Yes.
Okay, sir. Okay, sir.
And plasmids can go in at a very high efficiency, not only linear.
Okay. Okay, sir. Thank you.